Hi guys, it's Abby, and today I'm going to be talking about the genetics of the axolotl. It is a very special kind of salamander that is native to Mexico, and scientists are currently studying the role the genetics play in their really special abilities. They're able to regenerate their limbs, lungs, jaw, spine, even parts of their brain, and they're also very resistant to cancer, actually 1,000 times more resistant. So, scientists are studying them, hoping they can find things that are applicable to humans. And this is a picture of an axolotl. This is not the wild type is more gray. Their skin color follows Mendelian genetics. And it's not talked about in here because I didn't have space, but the genetic material of the axolotl is very special. They have the second largest genome out of any species. It is about 32 billion base pairs. And they also have the largest chromosomes ever discovered. But interestingly, the number of protein coding genes that they have is comparable to humans. They just have a lot of repetitive sequences and introns. So I'm going to start by talking about the p53 gene. In humans, the p53 gene is really important for preventing cancer. When it gets mutated, about 50 to 60% of cancers in humans are caused by mutations in the p53 gene. I added this graphic right here. So normally p53 is inactivated, binded to MDM2, as you can see right here. But when things like DNA damage, cell cycle abnormalities, hypoxia occur, it unbinds and the p53 becomes active. And from there it can go in two pathways. If the DNA damage is too extensive, it's going to go through apoptosis, which is basically telling the cell to kill itself. Or if it's fixable, it will pause the cell cycle and it will go through the DNA and fix it, and then allow it to start back up again. There's also some activation of other proteins. This is a very simplified version. And scientists have, well, this is one of the most studied genes that there is. And scientists have wanted to study it, but obviously aging plays an important role in cancer, and many model organisms like mice are not able to help scientists study aging. The axolotl's it can live up to 25 years old. And the p53 also plays a role in preventing cancer in the axolotl. It's also involved in limb regeneration, which I'll talk about later. And there are a lot of similarities between human and axolotl p53 genes. So by studying the axolotl p53, we could learn more about human p53. But a major difference that might have a role in the axolotl being 1,000 times more resistant to cancer is that there are some changes in the axolotl p53 DNA sequence that are found in the cancerous mutations in humans, but they have no effect on the axolotl. They're just a normal change. And another thing about the axolotl is they have extra copies of the p53 proteins that help resist cancer. And having extra proteins has shown that mice are more resistant to cancer, so it's a hypothesis that the extra proteins also help with the extra cancer resistance. And the p53 gene also plays a role in the axolotl's ability to regenerate limbs. Scientists found that when they turned off the p53 gene, the axolotl was not able to regenerate its limbs. And another thing they did that made the axolotl unable to regenerate the limbs was they took out the macrophages, which prevent intense scarring. And that intense scarring essentially turned off the gene. But once they reintroduced the macrophages, the regeneration genes were expressed again. And humans and axolotls have very similar genes. The key difference is how they're expressed and regulated. Human pathways prioritize that intense scarring. This is likely due to an evolutionary advantage. But scientists are studying how they might be able to have the genes expressed. Like the regeneration genes could be in human cells, they're just non-functional right now. And what the future applications of all this research? They're hoping scientists and medical professionals are hoping to learn how we can advance regenerative therapies and cancer medicine. And this shared evolutionary origin could provide a pathway for learning how humans could express those regenerative genes. It's likely very, very far away, possibly not even in our lifetime. But axolotl genes are able to regulate human genes. They work at different temperatures, but they are able to activate human genes. And they have already used oocyte extract from the axolotl to reactivate human tumor suppressor genes and stop breast cancer growth in humans. So even though a lot of this stuff is far away, it still has present day applications. And I just want to mention that the wild axolotls are facing critical endangerment, and all of this genetic diversity could be very important in learning about these treatments. So it's important that scientists do not ignore the endangerment of the axolotl. While we should focus on this, we should also focus on preserving that genetic diversity. And that's it for my presentation.